Can you give, okay, I have recording permissions, excellent, okay. And now I should share screen, share, and then, okay. Can everyone see the slides? Is that yes? Okay, cool.
Yeah.
Okay, awesome. And everyone can hear me, good. Thank you everyone for coming today, especially in these suboptimal conditions or optimal conditions, depending on how you look on it. My name is Nur Shahir, as mentioned before. I'm a doctoral candidate in the labs of Dr. Terry Fury and Dr. Shahzad Sheikh in the curriculum for bioinformatics and computational biology. Today, I'm going to be presenting on some of my thesis work involving intestinal inflammatory bowel disease, differentially affects intestinal region composition and aerotolerance profile of mucosally-adherent bacteria. So this is just a brief overview of the talk. First, I'm going to be discussing some of the background for my thesis work, and then I'm going to be discussing two of my projects, the first of which involves just Crohn's disease, the second of which involves looking across inflammatory bowel disease subtypes or IBD subtypes, and then we'll go to conclusion. Okay. So what is IBD? Well, IBD is a non-infectious chronic inflammatory disease of the gastrointestinal tract. It can take place anywhere along the gastrointestinal tract, but primarily is seen within the colon, commonly known as the large intestine, and the ileum, the terminal part of the small intestine. There are two primary subtypes of this disease, Crohn's disease, also known as CD, and ulcerative colitis, also known as UC. And let me see if I can put the, yes, okay. Okay, so there are multiple components in the pathogenesis of IBD. The common understanding of IBD as we know it is it involves a genetically susceptible host undergoing an inappropriate immune response to the host microbiota. And all these components play a role into how the disease develops and evolves. And the focus of my research, as mentioned by Terry, is the intestinal microbiota. So what do we know about the intestinal microbiota? Well, this is a figure on the left from the Human Microbiome Project. Each column corresponds to the composition of a stool sample at the relative abundance of various phyla seen in a stool sample across patients. Each column corresponds to a patient. The colors correspond to what phyla they belong to, and for those who do not remember the phylogenetic tree, phyla is like the highest level below bacteria. So there's bacteria, and then you have phylum, and these phylums are seen in these colors here. So there's, as you can see in this figure, there's a high amount of interpersonal variability, and these are all healthy samples. So even in healthy situations, there's a large amount of variability. That being said, studies have shown that there does involve 4 primary phyla that tend to be common across all patients: Firmicutes, Actinobacteria, and Bacteroidetes, and Proteobacteria. Okay. That being said, while there's high interpersonal variability, the function of the bacteria is highly conserved. So bacteria has a function. These are the same exact patients looking at their stool samples, only this time we're looking at the functional capacity seen within the bacteria in their composition. And as you can see, while there is like small little dips and rises in the types of functions that are seen, overall there seems to be a conserved core. Okay. So along with having various functions, bacteria has different relationships with oxygen. So normally so the general knowledge about bacteria is that it generates energy either aerobically through glycolysis or anaerobically through fermentation. But bacteria is actually more of a spectrum and much more complex. So bacteria some bacteria always generate energy aerobically and can only exist in the presence of oxygen. These are referred to as obligate aerobes. Some bacteria can only exist in oxygen levels that are lower than atmosphere, and those are often referred to as microaerophiles. Then we have bacteria that can generate energy either aerobically or anaerobically, and those are referred to as facultative species. There are other bacteria that only generate energy anaerobically, but can exist in the presence of oxygen. And there are other bacteria that they can only generate energy anaerobically, but they also just die if you present them in oxygen. So why do we care about oxygen and bacteria in relation to the intestinal microbiota? Well, this figure right here is a cross-section of your intestine, be it small intestine or large intestine. The outside bit is the epithelial wall. This rim right here is a cartoon version of our intestinal mucosa. The shapes refer to what type of bacteria, be it an OA, obligate anaerobe, or FA, facultative anaerobic species. And then this open space right here, we have the intestinal lumen, the open space within your intestine. Now, studies have shown that the oxygen gradient is decreasing radially, cross-section radially along the intestine. So we go from the epithelial wall to the mucosa to the inner lumen, you have a gradually more and more anaerobic space. Studies have also shown that the oxygen also decreases along the gastrointestinal tract. So in theory, it's generally known that the large intestine is a much more anaerobic space than the small intestine, but has not been proven through computational means. So how does this relate to IBD? So the figure we have right here is what we see in a normal healthy patient. So in a normal healthy patient in their intestinal microbiota, we have the intestinal lumen that's usually filled with commensal bacteria. These are bacteria that are usually beneficial to the host or at least do not cause any potential damage. We have anti-inflammatory bacteria that help mediate the immune response, as well as some pathogens. We also have this mucus layer in which you do see some bacteria in there, but not as much, the epithelial barrier, and then the lamina propria, where we have we usually get an inflammatory response. So inflammation normally is not bad. It's the way of your body protecting itself and reacting to injury. The problem in Crohn's disease and IBD, though, is that there is this chronic inflammation that characterizes the disease. And we also see the microbiota altered. We see an increase in pathogenic bacteria, as well as a loss of commensal and anti-inflammatory bacteria. And the mucus layer is also diminished somewhat, and pathogens are able to get through the epithelial barrier, which adds to the chronic inflammatory response. So as I mentioned before, there's a high amount of interindividuality seen in the intestinal microbiota. And this has caused problems in trying to generalize what we see in the microbial shifts that we see in disease. When you have a very highly variable population already, introducing disease, you're trying to separate what are potential confounders, such as diet or medication uses, versus what is actually due to the disease. So in disease, shifts in the microbiota are referred to as dysbiosis. Dysbiosis broadly just means a shift, but it's usually like a shift that is non-beneficial to the host. So for this case, I'm referring to IBD-associated microbial dysbiosis. And while there is a high amount of individuality, both in health and also in disease, studies have come to a broad understanding of what's happening. We know that there's a loss in microbial diversity, which I will explain shortly, as well as a loss of commensal bacteria such as F. praunici. We also see an increase in bacteria within the Proteobacteria phylum. In particular, we see an increase in the Enterobacteriaceae family, which is within the Proteobacteria family, Proteobacteria phylum, which includes pathogens such as E. coli. And we also sometimes, depending on the study, see presence of specific bacteria like Mycobacterium and Bacteroides species. So in order to try to understand what's happening in this IBD-associated microbial dysbiosis, Rutger Goss in 2013 proposed the oxygen hypothesis as a way of explaining the dysbiosis occurring in inflammatory bowel disease. And he noted that the things that we see, such as the loss of microbial diversity and commensal bacteria, are usually corresponding to the loss of obligate anaerobic species. Primarily, obligate anaerobic species are usually involved with processing short-chain fatty acids and other necessary functions that are needed. We also see an increase in facultative anaerobic species, and these are commonly seen in the family Enterobacteriaceae and proteobacteria and other pathogenic species, as well as the presence of aerobic species as well. And he proposed that this shift was why, what was occurring in the gut was why IBD is what was driving the dysbiosis seen in IBD. So this brings us to the primary challenge of our study, which is there's high individual variation in the microbiota, and this has limited our ability to identify consistent microbial signs associated with IBD. Which brings me to my project, also known as a fairly bold goal, which is determine consistent microbial signals associated with IBD. Okay. Which brings me to my first project, looking at the intestinal microbiota alterations within Crohn's disease. Okay. So we had some specific goals going into this study. We first wanted to see, does our patient cohort, first of all, emulate similar trends seen in prior studies? Basically, this is more of a sanity check that makes sure, like, at least our patients, our Crohn's disease patients, look like everyone else's Crohn's disease patients from a broad level. Next, we wanted to figure out, are there region-specific alterations in the mucosa-adherent microbiota in inflammatory bowel disease? And I'm going to explain what I mean by mucosa-adherent microbiota shortly. Are there disease subtype-specific alterations? And then, is there an in silico approach to investigating the oxygen hypothesis as proposed by Ritgoa-Guas? So, when we're looking at the intestinal microbiota, there are multiple ways of doing, but there's two primary ways. There's one looking at the stool and liminal microbiota, and there's looking at the mucosa-adherent microbiota. Your intestines are lined with mucus, so we're looking at the bacterial contents that are within that mucus layer. So there are pros and cons to choosing the source of your intestinal microbiota for your survey. When you're looking at the stool liminal contents, there is the benefit that there's an ease of use, and it's a non-invasive procedure. You just give them a bag or a sampling cup and say, have at it. The limitations is that it's an overview of the gastrointestinal tract. So you're not going to get that region-specific specificity, which is a problem, especially in Crohn's disease, where you have patches of inflammation that are non-contiguous. So you have regions of inflamed and non-inflamed regions. In contrast, while the mucosa-adherent microbiota is an invasive procedure, these are patients who have to go in for surgery to require these samples. The benefits are we're getting a region-specific snapshot of the microbiota. So instead of getting an overview, we can look at, what happens in the ascending colon part of the microbiota or in the terminal ileum part of the microbiota. And since we have this chronic inflammatory response as seen in Crohn's disease, the mucosal microbiota is also potentially interfacing directly with the disease, which is why we chose the mucosal-adherent microbiota for the focus of our study. So when we're doing a microbial survey, there are two different primary approaches. There is targeted sequencing, in this case, 16S ribosomal RNA sequencing, or a metagenomic approach. So metagenomics, there's some benefits to it. It's not just limited to bacteria. When we're referring to the microbiota, we're not just referring to the bacterial contents, we're referring to all microorganisms. So this includes fungi and archaea. And it covers also metagenomics, you're covering the entire microbial genome. So you're not just looking at the 16S gene, but also non-16S genes as well. The limitations, though, is that this is a very expensive procedure. And second of all, the mucosa is heavily contaminated by the host. So when you do metagenomic sequencing, you're sequencing everything. So as well as the microbial contents, you're also going to be sequencing the host. And in mucosa-based studies, that means around 90% of the reads that you're getting are going to be associated with the host. So you're throwing out a lot of data for an already expensive experiment. Contrast to 16S data, it only surveys the bacterial components, so we don't have an idea of what's the composition of the fungi and the archaea in this sample. That being said, the benefits are it's cheap, and cheap is good in many cases. It's easy to do, and it tolerates host contamination primarily because it's a targeted sequencing approach. So it's targeting the 16S gene, which is unique to bacteria. And thus, we chose 16S RNA as approach to our study. So for those who don't know or are not familiar with bacterial genomics, the 16S ribosomal RNA gene is seen right here. This is a secondary structure of what the gene looks like. It's very pretty. It consists of variable, hypervariable regions and highly conserved regions. And when we perform 16S sequencing, we're taking advantage of this. Why? Well, this is the same figure as before, only drastically simplified. The 16S gene consists of 9 hypervariable regions and highly conserved regions. So when we perform 16S sequencing, for this study we use the V1 V2 region, we're using primers to attach the highly conserved regions, since they're highly conserved, when we can create primers to basically stick onto those regions to perform sequencing. And then we can use PCR to amplify the region of interest. And for that, we use Illumina MySeq to generate paired end reads. So for those who don't recall offhand, genes have a 5' end and 3' end. So primer 1 is going to be going from the 5' end to the 3' end, while primer 2 is going to be going from the opposite direction. That's why we have these, they're referred to as paired end reads. So once we have these reads, we need to figure out how to get from these sequences, these like collections of A, T, C, and Gs, to defining taxa, because ultimately we want to know what's the microbial, what's the bacterial composition within these samples. And there are 2 broad ways of doing this. The prior most common way of doing it is operational taxonomic units, also referred to as OTUs. So if we pretend here that all these dots are reads, with OTUs, usually we perform this through some percent similarity, usually 95 or 97%. And we take a centroid and we say, okay, we have this sequence and we consider it a centroid. And we're going to look for all the sequences that are 97% similar. And then we're going to cluster them into clusters like so. And then we're going to claim that these are OTUs, these clustered of sequences. And these are a computational interpretation of a taxonomic species. But the problem is that when you have this approach, you're losing some of the potentially using specificity. While you have your centroid, and yes, these are like 97% similar, there may be cases where you have 2 sequences that are like a lot more similar to each other as opposed to anything else in the cluster. And then you're wondering, okay, should these really be clustered within this operational taxonomic unit, or are they actually distinct? So this brings us to another approach more recently proposed by Ben Callahan at NC State, called amplicon sequence variants. So the amplicon refers to the region that you amplified, and we have sequences from those, as mentioned before. Each of these are all sequences, but instead of clustering by percent similarity, we look at the data and infer error models to figure out when you perform sequencing, there's often errors. So trying to predict, okay, how many times is it slipping from A to a T or a C to a G? And let's try and infer probabilistically, what's the probability that a sequence is actually a unique sequence of its own, or it's something due to sequencing error. So you create an error model to use to predict, okay, actually these sequences are much more alike. And within our error model, it's highly likely that these 2 sequences are actually the same, just like sequencing errors, but these are not, they're not the same as this sequence seen here. And from these, we cluster them into what we refer to amplicon sequence variants instead. And one benefit of these amplicon sequence variants is that we can use these to, in many cases, accurately assign these sequences down to a genus or species level when we're going against a database. There are multiple databases of currently sequenced 16S genes across multiple bacteria. For this study, we used RDP, and for our classification approach, we used amplicon sequence variants. Okay, so after we get this data, we perform various analyses such as alpha diversity, also referred to as microbial diversity, differential abundance analyses, and these are the two common analyses that you see in the majority of microbiome papers. But then we also added a novel approach, which is the aerotolerance analysis, which I will discuss shortly. So, as I mentioned before, we care about diversity within our study and within the bacterial composition. Microbial diversity, or in this case, alpha diversity, for which we looked at in the study, is referring to two characteristics of a bacterial composition. This is something that we've taken from microbial ecology, has borrowed from general ecology. So alpha diversity is broken into two components, richness and evenness. Richness refers to the number of unique species seen within a sample. As we can see here, we have patient A, patient B, and patient C. The shapes refer to different bacterial species seen within that sample. As you see in patient A, it has three distinct bacterial species, while patient B has 4 distinct bacterial species, and while patient C has only 2. But there's also this concept called evenness, which is referring to the distribution of these samples, the distribution of the bacteria seen within a sample. As you can see here, while there are 3 distinct bacteria, the distribution is fairly even. You see 3 of each. As compared to, in contrast to patient B, you see there's a lot more of these blue triangles compared to the other bacteria. So there is this sample would be considered less even. And this combination of richness and evenness is what we use to define microbial diversity, or in this case, alpha diversity within our sample. So the next thing we did was an aerotolerance analysis. And this came to one of the first problems I noticed within doing this research was there is no existing database with aerotolerance classifications. So we went old school and did literature searches, mining literature by primary sources such as Burgess, the Prokaryotes, and then the International Journal for Systematic Microbiology. And from these, we used to classify the 20,000 plus taxa that we had got from our samples as accurately as possible, and then we normalized the data by patient to determine the relative abundance within the 5 categories of interest: aerobic, anaerobic, obligate anaerobic, and facultative. Okay, now let's talk about the study. So we had a patient cohort that we acquired from UNC, and from these we obtained mucosal biopsies. For this part of the study, we had 46 Crohn's disease patients and 23 non-IBD patients, of which we obtained biopsies from both the colon and ileum. Some, not from all patients, but some patients we had both, and some patients we only had a colon biopsy or an ileum biopsy. Just a reminder, colon refers to the large intestine, ileum is referring to part of the small intestine. And with these biopsies, we took samples from both inflamed as well as non-inflamed regions for our Crohn's disease patients. So the first question we wanted to see was, does Crohn's disease affect the diversity in each intestinal segment? Since we're using mucosal biopsies, we have the ability to look at region-specific changes. We noticed, like in previous studies, the colonic mucosa does undergo a loss of diversity in the presence of Crohn's disease. As seen here, we have the mean, we have the group sample size, as well as the mean, and if it's on the left, that means that group increased in diversity. If it's on the right, that means that group decreased in diversity, as well as p-values. And we see that compared to non-IBD patients, the colonic mucosa has a lot less diversity. We also looked at inflammation, which is a primary phenotype seen within Crohn's disease. And when we looked across all our inflamed and all our non-inflamed patients, interestingly enough, we saw that our inflamed patients showed an increase in diversity compared to our non-inflamed Crohn's disease patients within the colonic mucosa. But when we restricted to only matched patients, while we only had 3 for 3, we noticed that this difference disappeared. Next, we looked at the ileal mucosa, and similar to colon, it also undergoes a loss of diversity in the presence of Crohn's disease. We also noticed when we're looking at inflammation, unlike the colonic mucosa, the non-inflamed samples actually had higher diversity than the non-inflamed samples. The non-inflamed sample has higher diversity than the inflamed samples. But when we looked at our matched samples, samples that we had both inflamed and non-inflamed tissue from, it actually switched, where the inflamed samples had higher diversity than the non-inflamed samples. So next we looked within disease at tissue differences. So we wanted to figure out, okay, does the presence of Crohn's disease alter the diversity seen between the colon and ileum? So in non-IBD, we don't see much of a shift in the diversity seen between the two regions. And though the diversity is decreased in Crohn's disease, between the colon and ileum, you're still seeing similar levels of diversity. The next question we wanted to answer was, is there evidence that the oxygen gradient is being altered in Crohn's disease? So just a reminder of the oxygen hypothesis as proposed by Ritger-Ross. We see this loss of microbial diversity and loss of commensal bacteria. That's corresponding to decrease in oblique anaerobic species. We're seeing this increase in proteobacteria and Enterobacteriaceae species. That's usually corresponding to this increased presence of facultative anaerobic species. And the specific bacteria we usually see tend to be aerobic species as well. So first we did, with their air tolerance analysis, we looked at the relative abundance of our 5 categories: obligate anaerobes, anaerobes, facultative anaerobes, aerobic, and obligate aerobic species within the colonic mucosa in non-IBD and Crohn's disease patients. And we noticed that the colon, oddly enough, does not undergo a loss of obligate anaerobes in the presence of Crohn's disease, as you can see right here. While there is maybe like a little bit more variability within the colonic mucosa, overall, there doesn't seem to be much of a loss. Next, we looked at the ileal mucosa between non-IBD and Crohn's disease. And in complete conflict with the oxygen hypothesis, which claims that you're going to be seeing a loss of obligate anaerobes, in Crohn's disease, actually, it seems becoming more anaerobic in the presence of Crohn's disease, seen right here. We also notice that the variability seen within the ileum seems to be a lot higher. So then we looked between colon and ileum and non-IBD to figure out what are we seeing in a non-IBD state. And similar to what's known on a clinical level and more general knowledge, but it's not been shown from a computational perspective, the colon is more anaerobic than the ileum in non-IBD. And we also see more facultative anaerobic species and obligate aerobic species within the ileum as well. And this is in line with what we're seeing with the decreasing oxygen along the gastrointestinal tract. Interestingly enough, in the presence of Crohn's disease, when we're looking between the colonic and ileal mucosa, Crohn's disease, the ileum just seems to be adopting a more colon-like environment, while there is distinctly higher variability compared to the colonic mucosa. We are seeing this more of a sort of, not homeostasis, but sort of like more adopting more of the characteristics of the colon, at least in the presence of Crohn's disease from an aerotolerance perspective. So our next question was, okay, so what taxa are significantly altered in the intestinal mucosa in the presence of Crohn's disease, as well as in non-IBD, and do they reflect the diversity and aerotolerance results that we're seeing? Okay, so this figure is very intense. I'm going to slowly walk you through it. The x-axis is corresponding to the -10 log p-value, and the y-axis is corresponding to the log 2 fold change. So basically, if it's above 0, that means that taxa was increased in the presence of Crohn's disease. And if it's below 0, that means that taxa was decreased in the presence of Crohn's disease, i.e. CD depleted. The colors correspond to what phyla that the taxa comes from. If you remember previously, phyla is like the first level right below kingdom. And it's like the first way of defining a bacteria species. The shape of the dots corresponds to what category they came from in our classification. So whether it's an aerobe, an anaerobe, facultative anaerobe, obligate anaerobe, or whether we were unable to classify it. And then finally, the size of these dots, as you can see, are variable. The size of these dots are is the scaled average relative abundance of that taxa within the region that it's increased in. So in this case, the size of this dot corresponds to the mean relative abundance of Bacteroides fragilis as seen in the colonic mucosa of Crohn's disease patients, while the size of this dot here, this Faecalibacterium, which is a bacteria normally associated with health, the size corresponds to the mean relative abundance of this taxa within non-IBD patients. So as you can see by the copious amounts of filled in circles, the alterations we primarily see in the colonic mucosa between non-IBD and Crohn's disease are primarily alterations in obligate anaerobes. This is in line with our Aerotolerance analysis, where we noticed there wasn't really a loss of obligate anaerobes. And in this case, in this figure here, we're seeing more of a swapping of particular obligate anaerobes. We also performed additional analysis to identify other potential dysbiotic taxa that seem to be primarily in our Crohn's disease cohort. And these included bacteria such as Bacteroides vulgaris, as well as E. coli, which is has been shown to be associated with Crohn's disease. But interestingly enough, when we looked at the mean relative abundance of all these changes, they actually only encompassed about 8%. So while we're seeing a lot of shifts, they're not impacting the composition of the colonic mucosa very much as a whole. Okay. And then we looked at the ileomucosa between Crohn's disease and non-IBD. As we can see here, we're seeing a lot more of these filled circles, these obligate anaerobes that seem to be enriched in the presence of Crohn's disease in the ileomucosa, as well as pathogenic proteobacteria such as Klebsiella and other Enterococcus species. But we're also seeing, we're still seeing a loss of obligate anaerobes, but these are at a much lower relative abundance, and these are also common. These obligate anaerobes seem to be more associated with health, like Lactobacillus species, the Faecalibacterium. These are common bacteria that are often lost. These are often considered commensals. As you also can see, there are more taxa altered, and it does seem to primarily involve obligate anaerobic species as well as these filled diamonds, facultative species. And interestingly, unlike the colonic mucosa, these seem to take up around 46% of the relative abundance. So there's a lot of change happening as a whole to the composition of ileomucosa in the presence of Crohn's disease. Okay, so next we wanted to see, okay, what's happening in a non-IBD state between the colon and ileum? And as seen in the aerotolerance analysis as shown before, the ileum is a much more aerodiverse environment than the colon. We have more facultative anaerobic species, as well as some obligate aerobic species, as seen by these X's, by the triangles and X's of aerobic species, and we see more facultative species as well. But we also see obligate anaerobes, so not as many as we see enriched in the colon. And this is reflective of the Aerotolerance analysis that we performed earlier, where we saw that the colon was a much more anaerobic environment than the ileum. Next, we looked at Crohn's disease patients between the colon and ileum. And first of all, we're seeing a lot less changes between these two regions, but also of the taxa that are altered, they tend to be primarily from the Clostridia class. So lots of these green dots correspond to the Firmicute phylum, and the Clostridia class is seen within the Firmicutes phylum. And these are the primary taxa that seem to be altered. And there also seems to be an overall less distinctiveness between the colon and ileum, which seems in line with the aerotolerance analysis, where we're seeing the ileum environment from an aerotolerance perspective becoming more colon-like. So in summary, diversity is lost in the presence of the presence of Crohn's disease in both the colon as well as the ileum. But between region diversity is not significantly altered. And our aerotolerance analysis fails to support the oxygen hypothesis as proposed by Richard Ross. The relative abundance of obligate anaerobes in the colonic mucosa seem to be unaltered between Crohn's disease and non-IBD. And in the ileal mucosa, the the relative abundance of obligate anaerobes seems to be actually increased in the presence of Crohn's disease. And our differential abundance analysis also seems to reflect the results seen in the aerotolerance analysis. Okay. So next I'm going to be talking about some of my more recent research. The previous mentioned research has actually just been accepted for publication, but this is more recent research. So this is looking at the intestinal microbiota alterations across IBD subtypes. So as before, I have a patient cohort from UNC, but this time we're looking at mucosal biopsies only within the colon. And from this, we have 20 UC patients, 15 CD patients, and 16 non-IBD patients. And we have both non-inflamed and inflamed tissue. So just a reminder of the differences between UC and CD. While both UC and CD are subtypes of IBD, their presentation is radically different. Ulcerative colitis involves contiguous inflammation along the gastrointestinal tract. So as you can see here, some just involve the rectum, while other parts involve more of the colon, while to entire inflammation of the gastrointestinal tract, referred to often as pancolitis subtype of UC. In contrast, in Crohn's disease, you're seeing more of a patchy inflammation. So instead of the contiguous lines of inflammation that you're seeing in UC, you're seeing regions of inflamed tissue and non-inflamed tissue. And those are the two defining characteristics between a UC patient and a CD patient. So first question we wanted to know was, does diversity behave the same across IBD subtypes? So we noticed that actually when we're looking in both UC and CD patients, instead of them both losing diversity at the same amount, it seems that there's actually more of a decreasing gradient, with non-IBD patients having the highest diversity, followed by UC, followed by Crohn's disease. And this is in line with some literature where either UC patients are undergoing a great loss of diversity, and some other UC patients seem to maintain a similar diversity compared to non-IBD controls. Our next question was, does aerotolerance difference between IBD subtypes? So as mentioned previously, as we saw, at least in the colonic mucosa, there's no significant alteration in the aerotolerance between the colonic mucosa between Crohn's disease and non-IBD. So initially thought that UC would behave in a similar fashion. Interestingly, UC undergoes a loss of obligate anaerobic species in the presence of UC. But also, unlike Crohn's disease and non-IBD, there's also a lot more variability in the relative abundance of obligate anaerobic species compared to Crohn's disease and non-IBD. As well as a wider range in the relative abundance of facultative species. And we thought this was a bit odd. We also saw an increase in facultative species compared to non-IBD, as well as an increase in aerobic species compared to both non-IBD and Crohn's disease. But we found this high variability a bit odd, especially when looking at Crohn's disease, where there was not as much variability. So we decided to put this on a scatter plot. So each dot corresponds to a patient, and the y-axis corresponds to the relative abundance of obligate anaerobes seen within that patient. So as we can see here, the majority of Crohn's disease patients are above like the 50% amount. There's like this one person, and we did check for medication issues, and it was just seemed that this person just has a lot of facultative anaerobic species. We also noticed in non-IBD, we see this nice anaerobic space that's characteristic of the colon. But within UC, interestingly enough, it seems that these patients seem to stratify into patients that have a high amount of relative abundance of obligate anaerobes in line with along with a non-disease colon, And then we have another subset of patients that have a very low abundance, or very low relative abundance of obligate anaerobic species in their composition. So we decided to split these patients into subgroups and reperform our aerotolerance analysis. As you can see here, it does seem that our UC patients seem to stratify in the relative abundance of obligate anaerobic species that we see within our cohort. We performed a test looking at the overall distribution, and we compared these patients to our high group, or our UC health-like patients. They seem to have a fairly similar distribution from a aerotolerance perspective, in contrast to our low patients, which are radically different. So next, and I'm only showing some of the results looking at the patients in the high group versus patients in the low group for UC. As before, the colors correspond to what phyla these species come from, and the size of the dots the shape of the dots correspond to the aerotolerance category, and the size of the dot corresponds to the mean relative abundance within the category that it's enriched in. As you can see here, in the high abundance group, we tend to see a lot more obligate anaerobic species. You see some facultative aerobic species, but overall anaerobic. We also see bifidobacterium, which is associated with barrier health as well in the intestine. In contrast, in the low patients, we're seeing more of the pathogenic bacteria such as Klebsiella and Enterobacteriaceae, which are within the Proteobacteria phylum. So in conclusion, within IBD, there seems to be a decreasing gradient, with non-IBD patients having the highest diversity, UC patients undergoing a loss of diversity, but CD having the deepest loss of diversity in the colonic mucosa. UC patients also seem to have a higher variability in the relative abundance of obligate anaerobes in the colonic mucosa. And UC patients seem to split into a high and low obligate anaerobe composition stratification, with the low type in line with the oxygen hypothesis. We're seeing this depletion of obligate anaerobes as well as an increase in facultative species and aerobic species, while the high subtype is more in line with our Crohn's disease patients, which contradicts the oxygen hypothesis. So in conclusion, alterations in the mucosa-adherent microbiota are region-specific in CD, and it's only because our study involved mucosal biopsies that we were able to get this type of resolution. And this implies that it might be while it is harder to obtain, it might be beneficial to have more mucosal-driven studies. Previously, there's been primarily colonic mucosal studies and a few ileum ones, but given the characteristics of IBD, it seems essential that we have more mucosa-based studies to get a better idea of what are the alterations and better understanding of them as well. And we have the oxygen hypothesis, which is saying that we see a loss of obligate anaerobes and increase of facultative species in the presence of Crohn's disease. But it seems that Crohn's disease does not follow this profile within the colonic mucosa nor in the ileal mucosa, but UC seems to split into two varying subtypes that we still need to explore more, as well as look into larger cohort studies to see if this is actually a replicable signal. Okay. So I would like to thank my labs, both the Fury lab and the Shake lab, as well as my committee, as well as my funding. I have a few more acknowledgement slides because as some would say, a picture is worth 1,000 words. And I have been extremely fortunate to be here at UNC and to conduct my research here. These are pictures of my labs, both current and present people. I'm also very grateful for the people who are heading the department, heading the curriculum for bioinformatics and computational biology, including Will Valdar and Tim Elston, as well as my committee as seen here. I'm also very thankful for TIBS and IMSD and the other resources offered through the graduate school at UNC. They're honestly very impressive, as well as very helpful and very approachable. There have been many times when I've just run into the office and just collapsed. as well as just a small subset of my friends that I've made here through the computational program. I've also made friends outside the computational program, but I only have so many pictures because I'm bad at taking pictures of things other than food. I also like to thank John Cornett, also known as the Student Service Administrator, also known as the only reason why I have my paperwork done in time, and just honestly, he goes above and beyond. So honestly, thank you, John, so much for all the assistance you've given all the years. This slide is not enough to say thank you, but like you get your own slides, so yay. I'd also like to thank my family, both people who are no longer with us, as well as people who are still with us, either close to me or far away from me. But especially I would like to thank my parents for always being amazingly supportive and also just continually believing in me, even when I didn't believe in myself, and just being a constant source of support. I'd also like to thank my friends, and this is only a small segment of my friends who are thankfully able to actually attend, be the fact of, while we're in the time of Corona, it seems we've managed to get more closer, and thank you. And now I am open to questions, so.
Okay, so as I said, if you would like to ask a question, I know there's this raise your hand feature, which I don't honestly know exactly how it works. But if you do that and I see it, I can switch you on and let you take a question, or else if you send me a note through the chat that you have a question, I'll unmute you and let you ask. So questions, anyone?
Sharif Farag has a question.
All right, go ahead.
Hi, Nur. Great talk.
Thank you.
I just have this one question. So obviously, this is a very complex disease, and the data is pretty complicated. But the question that I have, it seems in your findings, you found that in UC, it is associated with less anaerobic bacteria, right, than in non-IBD.
We saw that the UC patients seemed to split into two potential subtypes based on aerotolerance, one of which had their obligate anaerobe composition maintained, which the other group, they seemed to undergo a depletion of them, yes.
Yes, so the question that I would have, can we use like anaerobic specific antibiotics to basically to target the high increase of anaerobic in these areas, and that this would help or actually increase the diet of aerobic bacteria like probiotics? Would this actually have any sort of impact? What I'm trying to say, are these anaerobic and aerobic bacteria in these regions, are these just statistical associations with the disease, or actually it's causality, like it's proven to be?
So currently, all the stuff I presented are association based. We do know from prior studies, prior review articles, that the composition of the intestines are predominantly obligate anaerobes, and that's good. That's good. So I sense what you want to say is not have antibodies targeting anaerobic bacteria as much as targeting aerobic bacteria. Would I be correct?
Right. Yes.
Unfortunately, regarding the antibody part, I am not entirely certain. I know that our mucosa does have antimicrobial peptides, but I'm not certain about the ability to create antimicrobial peptides that are targeted towards aerobic bacteria.
All right. And sorry, the last question would be, if there is a very clear line of treatment for Crohn's disease or ulcerative, or it's still it's a cocktail of things that
it is a cocktail of various biologics, anti-TNFs, diet changes, and so forth. The interesting thing about, well, not interesting, but sad thing about IBD is in general is that these are chronic diseases. They are not curable. The only quote unquote cures that exist for UC In the severest cases, they excise the entire colon, or in some cases, they can excise part of the colon, or or they can excise it and then form a sort of pouch with the small intestine. Which is why in some patients with UC, you see these colostomy bags because they don't have a rectum. Or in some, but the J-pouch procedure is a way of avoiding that end goal. In IBD, though, there is no cure as well. It's also treated with a similar cocktail of biologics, anti-TNFs, 5-ASA, and diet and so forth. Sometimes probiotics, but probiotics are kind of like a mixed bag. But the defining thing with IBD is you often reach a medically refractory state, and with Crohn's disease, you often reach a medically refractory state. And the Crohn's disease patients that we had were patients that had reached that medically refractory stage where none of the therapies were working or having any change. So in that case, then they excise the diseased part of your colon and ileum, and hopefully that fixes the problem. The problem is around 80% of patients who have to undergo surgery, within 20 years, the disease recurs. So treatment is still very much a work in progress.
All right, so we have a few other people that have questions. So Andrew.
Hi, Nur.
Hello.
Awesome. Yes. Here's a new baby, Andrew the fourth. He also enjoyed your talk.
Did he? Okay.
I mean, he might have fallen asleep through part of it.
Okay.
But I enjoyed your talk, and I had one quick question, because I might be diving down this rabbit hole a little bit more. But did you ever consider diving down the rabbit hole of looking at kind of community relationships within the populations of bacteria? So whether or not they were cooperating or policing each other in order to decrease diversity.
And honestly, that is something I'm very fascinated, that we're actually very fascinated in, but we have not done that yet, and we'll see how much time I have left before I have to leave to actually start diving into that. But yes, that is a very, very valid question, because lots of studies previously had been focusing at the microbial composition and looking at them as individuals. But as you mentioned, it's a community. They're interacting with each other, either competing or non-competing. That's the reason why treatment for C. diff is often a fecal transplant, is because you go undergo antibiotics. It cleans out most of your natural microbiota because it's not specific, but you still have CD that is very robust, and then can take over everything because nothing is keeping it in check. Which is why then you have fecal transplants to try and sort of not cull, but sort of like restrain the expansion of C. diff. So yes, so we're very interested in community dynamics. I know we have a new postdoc who's very interested in biological networks, so I may try and like convince him to teach me some things so I can like maybe start putting at least a framework down for it.
All right, Wes.
Is a great segue into my question. Has there been much research on fecal transplants in Crohn's and ulcerative colitis?
Yes, they don't work. At least for Crohn's disease, they don't work. UC, I think, is more mixed offhand, but I don't recall. But Crohn's disease, definitely no.
All right, and Victoria.
Hi, Nur, you did amazing. My question, kind of going off all of this, is, so you said most of the population, the populations you sampled for these patients had reached that refractory state. Have you guys looked, or was there a thought of looking at these like different microbiotas in patients with different stages of Crohn's and IBS, as far as like maybe like disease prediction or disease staging or anything?
Yes, there are studies that are looking into that. The most well-known is the Giver study, which is looking at a pediatric cohort. The problem is that Crohn's disease and UC and IB in general is very hard to diagnose. We don't have any molecular subtyping or markers for it. So first of all, when you get a patient with Crohn's disease, often they've had it for a while, so you aren't seeing the initial disease initiation stage. That being said, there are studies where they're looking at patients with quote unquote active Crohn's disease versus quiescent Crohn's disease, but we're still trying to understand the general progression of Crohn's disease and IBD as a whole, and sort of trying to figure out how do we find these patients before they get it.
All right, I think we have time for a couple of more questions. So Hillary? Let's see. Yeah, there we go. Okay.
Really great talk. My question was on the second project, and I wanted to know if there were any further differences in the cohorts between the obligate anaerobe high versus low populations. Were there any other clinical markers that showed any differences between them other than the different bacterial loads?
We actually looked in
relative loads.
We actually looked into that because our first question was like, okay, maybe this is just a side effect of the medication. And we determined there was no association between what group they were in and the medications that they were on. So that we're like, oh, that's weird. Okay. We also looked at the phenotypes. The majority of these patients are pancolitis patients. So there wasn't any distinctive bias towards the presentation of UC either. So we're still trying to figure out why that is.
All right, and one last question from Damien, who says that it will be a short one.
Yes.
Hi, Nur. Great talk. Do you know anything about sort of the host genetics for IBD and whether sort of the human variation associates with genes that create the sort of strange oxidative environment that ends up pushing the microbial communities to dysbiosis?
So I have a broad understanding of the genetics involved in disease. It's, as you can tell, it's a non-Mendelian disorder. And through GWAS studies, we have determined around 230 some odd loci associated with both with IBD, and some that are just associated with Crohn's disease, and some that are just associated with UC. So there's like not one nice gene per se. That being said, many of them tend to involve immune regulation, which makes sense since the big thing about Crohn's, the IBD, is that there's a dysregulation of the immune system. How it relates to the oxidative state, I do not have that information at hand.
Thanks.
All right, great. Well, I'd like to thank everyone for coming and listening to Nur's presentation and supporting her during this time. And at this point, we are going to, our dissertation committee is going to exit to a different Zoom meeting to go to the next part of the defense. But I hope everybody stays safe and well, and thanks again for coming. All right. Bye.
Bye.